Hello science fans, and welcome to SHE-ensya. Today, we're going back to basics and continue our discussion on evolution, but this time focusing on the changes that are happening inside and outside of our cells that are related to evolution. Now, we started our discussion on evolution on how animals have existed on planet Earth for about 500 million years. We then walked through the history of animals based on the Earth's 4 known eons. We then dove into the major extinction events that drove evolution, starting with the Ordovician Silurian extinction that happened 440 million years ago. From there, we looked at the confusing Devonian extinction that happened 365 million years ago, where many tropical marine species went extinct with unclear reasons. We then went through the Great Dying, the Permian-Triassic extinction that happened 252 million years ago, which comprised the biggest extinction event on Earth that was caused by the eruption of the Siberian traps. We then looked at the Triassic-Jurassic extinction that happened 210 million years ago, which led to the extinction of dominant vertebrate species on land that allowed the dinosaurs to rule the Earth. And finally, we discussed the dramatic Cretaceous Paleogene extinction that happened 65 million years ago that led to the extinction of the dinosaurs because of a massive asteroid strike. What's interesting is that throughout history, we see animals changing in form across time, usually sped up by the mass extinction events. But as scientists, what evidence do we have that evolution is really happening and that living things are related to each other? Well, we have the DNA. First isolated as a chemical in 1869, DNA, or deoxyribonucleic acid, is an acid and alkali-soluble, phosphorus-rich compound that is largely found in the nucleus of cells, but also in our mitochondria and the chloroplast of plants. Experimentation with bacteria and viruses have shown the transformative powers of DNA, and it has since been declared as the blueprint of life as early as the 1950s. This amazing double helical molecule has functional segments called genes, which code for molecules called proteins, which eventually become responsible for our physical characteristics, from our looks to our metabolism, and eventually even influencing our lifespan. Now, if you're interested to learn more about the DNA, its discovery, its dramatic history, and intrigue, please check out the video that will pop up over here at the end of this one. Now, since DNA is the blueprint that is the basis of our physical appearance and function, changes in the DNA, known as mutations, aid in evolution. There are 2 types of mutation that can happen to an organism dependent on which cells undergo the mutation: somatic or germinal. Somatic mutations, as the name suggests, are mutations that happen in somatic cells. Somatic cells are mature functional cells all over the body, from your eyes, to your skin, to your liver, and even your kidney. These cells eventually mature and die, and so the changes in their DNA would not get passed on to the next generation. Germinal mutations, on the other hand, are those that happen to our germ cells, basically our reproductive cells like the egg and sperm. And since germ cells, upon fertilization, form a new organism, mutations that happen to them get passed on to offspring and move from one generation to the next. Now, there is quite a variety of changes in the DNA that can happen, whether it's somatic or germinal. For example, one kind of mutation can occur through substitutions where some nucleotides are replaced by others. Substitution mutations can be silent, and this is when the final protein product of the gene is not changed. Silent substitution mutations can happen because the genetic code is redundant, and multiple combinations of nucleotides called codons can code for a specific amino acid, which are the building blocks of proteins. But a change in physical characteristics can occur if a missense mutation is what happens. In a missense mutation, the substitution in the DNA results to a counterpart change in an amino acid in the final protein. And you can also have a nonsense mutation that, despite its name, can also cause dramatic changes in the physical characteristics of an organism. Because in a nonsense mutation, the substitution shortens the final protein product because of the creation of an early stop signal. Now, so far, we've just been talking about substitution mutations, but another kind would be insertion mutations, and this is when one or more nucleotides are inserted within a gene. When the number and location of where the nucleotides are inserted results to a shift in the reading frame, this is called a frameshift mutation. A reading frame is how an mRNA, which is based from the DNA, is read by our cells' ribosomes in order to create proteins. The message from the DNA is basically interpreted 3 letters or bases at a time called codons, which are then equivalent to 1 amino acid. But when the number and location of insertion does not shift the reading frame, then this is called a non-frameshift mutation. Frameshift insertion mutations have greater potential to create dramatic changes in the physical characteristics of an organism because they often create a whole new protein. Whereas non-frameshift mutations have lesser impact because they often result to a very similar protein with only a few or even just 1 amino acid difference. And then we have your deletion mutations, which is when one or more nucleotides ghost the rest of the gene. Similar to insertion mutations, there are instances when the deletion results to a frameshift mutation. But when the location and number of nucleotides do not change the reading frame, this would result to a non-frameshift deletion mutation. Another type of mutation is when chromosomes, or parts of them, are added, missing, or changed. These are called chromosomal mutations, and there are 4 general types. Chromosomal deletions are when a portion of the chromosome is lost. A chromosomal translocation is when a fragment is transferred to a different portion of the chromosome or a different chromosome altogether. Chromosomal duplication or insertion is when a portion is cloned and inserted within the same chromosome, resulting to a larger chromosome. And finally, chromosomal inversion is when a portion is flipped and thus inverting the content. It is very important to note that mutations happen randomly, with their occurrence increasing in probability if you are continuously exposed to mutagens such as UV radiation from the sun, stress, or even some toxic chemicals. And when you get a mutation, more often than not, they are either removed by your body or properly corrected. If not, you don't immediately evolve. Sometimes you get sick, but most of the times, the physical change is too minimal to be observed. A mutation can lead to evolution if it is retained within a species across generations. And an environmental change makes the counterpart physical characteristic beneficial to the organisms who have them. For example, the giraffes that we see now have very long necks, but this wasn't always the case. There used to be a time, if we look at the fossil records and also the genetic data, when not all members of the species had long necks. The evolution of the long neck of the giraffe was because of generations of repeated natural selection. You see, with extreme changes in environmental conditions like global warming or global cooling, brought about by volcanic eruptions, asteroid strikes, or even human pollution, a giraffe with a long neck would have a greater advantage or a higher chance of survival than their short-necked counterparts because they would be able to reach food basically leaves at the top of trees, faster and more efficiently. Of course, it is possible that in a parallel universe where plants or trees evolve to be short and closer to the ground, short-necked giraffes would have the greater advantage. Our relatively new understanding of the DNA and the availability of genetic technology have made the study of evolution faster and more accurate. But scientists have been studying evolution for hundreds of years. What evidences and observations did they have back then? Well, for one thing, scientists were able to study and compare the early developmental stages of different animals. This refers to the field of embryology, which studies the different changes occurring in our bodies from the moment we are conceived until we are born. What scientists were able to see in their studies of the embryos of different animals is that even organisms that are distinctly different share common features as embryos and fetuses. Of course, over time, different animals develop distinct characteristics that differentiates them from other animal species. But the common appearances and structures at the earliest developmental stages hint at a common ancestor or origin. Scientists were also able to observe similarities in adult structures, such as how the bones of our arms and legs are very similar to those of a horse, cat, bird, or even a whale. These similarities are called homologous structures, similar physical characteristics of different animals with a common ancestor. Of course, not all structures that are similar across organisms imply an evolutionary connection. Some function the same but have different structures, such as the fins of a turtle, fish, or penguin. Similar body parts that function the same way but do not indicate a common evolutionary origin are called analogous structures. Now, both homologous and analogous structures imply evolution, just two different kinds. Homologous structures imply the existence of divergent evolution. Divergent evolution is when two or more species evolve from a common ancestor to develop unique characteristics that is more appropriate for the habitat that they have finally found themselves in. While analogous structures imply convergent evolution, which is when two or more species with different ancestral origins develop similar characteristics because of the habitat or ecological niche that they found themselves in. The study of evolution is such an exciting field. With new techniques and new discoveries, we get to learn more about how life finds a way to expand and flourish no matter where it goes. And that's it for our discussion on the evolution of animals for our Back to Basics videos. At least for this season anyhow. But just remember, the physical changes that accompany evolution is rooted in counterpart changes in the DNA called mutations. These changes are brought about by completely random events of mutation that can be corrected by our bodies, make us sick, or even go undetected. And evolution can happen when mutations are allowed to persist in a species and result to significant advantages for survival during hard times caused by environmental change. And even before DNA-based technology, scientists have been studying evolution through embryology and by looking at homologous and analogous structures in animals. Thank you so much for joining us in our Back to Basics video for today. I hope you were able to learn something about the changes inside and outside the cell related to evolution. If you did, I hope you can give this video a thumbs up and also subscribe to our channel. If you have any comments, questions, or suggestions, please don't hesitate to message me, your resident Filipino scientist, in the comments section below. Thank you very much, and see you around!